yield on the back end of the hybridization and capture to feed that forward into the MockSeq library prep. So we should be getting those reagents shortly now, and we'll have something to report back on hopefully in the next month or something.
Nice. What kind of N50s were you seeing with those original runs, the first runs?
With the original runs, yeah, everything was coming in around the 15 kb length. So we had the concatenamers were really nicely sized, and it was really uniform.
Okay.
Yeah.
Hi, my name is Saloni, and I'm from Takara. So I'm just curious. So when you say for your MassSeq, you had a lot of like off-target effects with the DT priming, like it is DT priming, so it's going to be whole transcriptome. So when you say off-target, like what? I was just curious.
Just reads that were not assigned to any specific transcript. They were potentially like intronic reads or something like that.
Okay.
And I guess this is a problem that's inherent to that approach in general. So I sat through a webinar on a Oxford nanopore based approach where they were basically saying the same thing, when they tried just global sequencing of poly T primed cDNA. They got a bunch of junk reads that they couldn't really do anything with, and so they basically pivoted to a targeted approach as well.
Okay, good to know that. And then I know that somebody also mentioned that the concatemer is 15 kb, but like I'm just curious, for each insert, like how long can you, like what is the individual fragment length that you can get?
I can look up the length really quick of our the segmented reads for those runs. Give me a couple minutes here.
I have a quick, while you're looking that up, Alexander, I just want to use this time. Judy, I know you mentioned adaptive sampling. Have you been using, like, what kind of organisms? Any just human or other things besides human?
Mouse and
does
rat.
one work? Mouse and rat. Does one work better than the other, or is it all pretty?
It's worked quite well. I mean, we had we're using the Promethean, and we had upwards of 40x coverage, I think, on the transgenes that are on the. We're targeting chromosomes, actually, not specific transgenes. We found that the enrichment is about the same. So once we found the transgene on a different flow cell, then we would target the chromosome that we've identified it on. Stuff, but yeah, we got really good enrichment. We were very happy.
Awesome. Anything? Well, I know I'm gonna come back to you, Alexander. I know you're looking, so I don't want to make you think that I'm like moving on.
No worries. I'm clicking through all of our.
Gotcha.
Okay, here we go. Our mean length of our segmented reads was 839 bases. Yeah. And for just, I mean, that's for one of the 8 samples we ran, but they were all pretty much in that ballpark area. And let's see, we got 25 for this particular sample. We had 25 million segmented reads, which is not bad for, that's pretty much in the ballpark of what you might expect for like a transcriptomic, like RNA-seq for a library. My take on the whole thing is like that's probably not nearly enough. Well, it's definitely not nearly enough for like global isoform analysis. So that was another reason that kind of drove us towards this targeted approach, is to get sufficient read depth to for the genes that the investigators really care about to do a meaningful analysis.
Right. And I mean, like the fragment length seems to be like smaller, right? If you want to try to capture the full length, all isoforms.
I think that's kind of inherent to the 10x library prep protocol. Their polymerase is just going to fall off after so far.
Okay, now I was just curious. Thank you.
You bet.
I have a question again too. I've also came prepared with questions though, in case there was something below. But I'm also just curious about how success were any Promethean or ONT owners about like kind of the direct RNA, if anybody's had like massive success or if it's been a massive flop in your hands. Ours, it's been kind of so-so. So,
Yeah.
yeah.
Our initial run was a massive flop. So and we have not gone back to it, but we have a new faculty member on campus, Kin Fei Ao, who's actually an expert in RNA modifications and has experience with RNA, direct RNA.
Cool.
So we're kind of waiting for the new kits to come out to
yeah.
get a little bit more involved.
Yeah, I hear you. Any other like burning questions? Like high molecular weight DNA extraction, QC.
I have a question actually, and I've had a couple of investigators approach me for HLA typing services, and that's something we can kind of do somewhat routinely on an Illumina sequencer, but I think it's really apt to long read sequencing. So I was curious if anyone had, I looked at PacBio. I know that PacBio has a suggested protocol for that, and there's a library kit through GenDx that was recommended on their website. But if anybody else has any sort of maybe more home baked approaches or other commercially available kitted approaches for generating those libraries prior to PacBio sequencing.
I personally have it. We kind of got it. It's like on a little back burner of ours. Like if a next spot opens up, we'll probably hop into it, but I haven't done it yet.
I guess I'll just say we do have some HLA genes in a couple of the twist panels. So that's an approach you could take. Just having a custom panel with twists would be something that could work.
Sure. Okay.
But
and
yeah, the
GenDx
there... kit is a nice one too. That does, I think, 11 of the class 1 genes.
Yeah. Is that their, like, when you say that the twist panels, is that they're off the shelf, like long read panels that they have?
Yeah, so there's 2 off the shelf panels. There's the dark genes panel, and that has, I think, 2 or 3 HLAs in there. And then there's the Twist Alliance long read PGX panel, which I think has 4 genes. Okay. The gene lists are on their website.
Yeah. Okay.
Yeah, but neither of those are like a comprehensive HLA panel by any means.
Great, thank you.
I have a question on the PacBio review system. So with the review, the non-hi-fi, the non-CCR sequencing is turned off, right? Only hi-fi sequencing is available, and that's as far as I know, that's the case, right, Molly or Sarah?
Sarah, correct me if I'm wrong, but I think yes, like the subreads are no longer being.
Yeah, it's not like the main data type. We do have like a separate file called fail reads, which has sort of a subset of the subreads that don't meet the quality requirements for hi-fi. So some of that data are available, but we're trying to shift folks to just focusing on hi-fi because we think it's just a much easier file type to work with, and the file size and computational footprint is much lighter with hi-fi.
What about the some situation that you want long read, not necessarily very high accuracy, right? Just you want to get very long reads. But you can compromise on the sequencing accuracy side. So I think there is some application there. So I think if you focus only on the hi-fi, then you miss cases like that. I mean, for users, I don't know whether you miss that part. For example, Molly, do you want to have that non-hi-fi on the review?
I mean, so we actually don't have the review. yet. But like I know that I think we've had discussions with some current customers who still do utilize their subreads, and so that's a good point of why I'm thankful that we still have our two SQL twos. So I, it's kind of it was a little I was a little torn. Like I mean I love hi-fi data. Like I think that's pretty great, but like I also like I think there are some clients just based on our diverse clientele that do need those subreads and do like to, take a gander. So
Yeah,
yeah.
you're good. You're good. You have the 2E there, right? Still running.
Yeah, we have two twos running. So
Yeah.
Yep.
So you can solve those problems. I'm thinking, for new users, if we just have one review, then for those we cannot take those projects.
We're closing in 23 seconds. That's not much of a warning. Oh my goodness. Okay, well, I'm gonna get that link for you guys, and I'm sorry I recorded a little bit late, but I think this was good. I think this was, I mean, for being the first time, like we're all like, what's happening? It was great seeing all of you, and I hope I did the session justice. Bye.
Thank you.
20 minutes is not enough, man.
Yeah,
Never enough.
Not enough time.
yeah. No, the discussion's just going, and I think this is fantastic. So we have, I'm gonna we can do the recaps that we've done in the rooms, or we can go back and do another 20 minutes. We have 30 minutes left in this session, right, Ken? Yeah.
We have as much time as the group wants, Kevin.
Yeah. So we can go back and reopen the rooms for another 20-minute session.
So Kevin, may I suggest that we go back to the same rooms for 15 minutes and then recap for 15 minutes so that people, when they go and watch the videos in the other rooms, if they watch the videos in the other rooms, at least will have some idea as to what's being talked about, can choose for themselves what's important for them.
Yep.
So they can just open it up for another 15-minute session. You can stay with where you were at, or you can jump to another session.
Oh, welcome back. I've missed you guys. I know we were talking about hi-fi versus subreads. Does anybody else have any experience on the need of subreads versus not?
I'm curious what like specific applications you really need the subreads for. I'd be interested to hear.
I would have to ask my clients because I don't do any analysis. They're just like, where's my subreads? And I'm like, okay, here they are.
Yeah, it's hard to know if it's like the pain of just sort of switching and updating to a new data type, or if there really are key applications where it's important to have longer reads. Because I know when we were initially, developing hi-fi reads, we did a lot of work comparing de novo assemblies with hi-fi versus longer CLR reads, and generally the results were better with hi-fi even when they were shorter because the quality seemed to help.
Yeah, I don't have. I did want to ask, and this is a tangent because we're gonna go to an ONT question now. Well, first, does anybody still I mean, if you're a core, do you offer Sanger? If you're not a core, do you still do Sanger?
No,
We do.
We don't.
Oh, sorry, Kirsten, I think.
Sorry, yeah, we're at UVM, Vermont.
Oh, Vermont. Okay, gotcha.
Yeah,
I saw
I'm a
the yellow
Scot.
pop up, but gotcha. You were Scott. Cool. Yeah. Is it like a lot of business for you, or?
We run it 3 days a week is what we offer. It's not always full 3 days a week, but sometimes it's 8 samples at a time.
Yeah, I hear you.
Right.
I was just going to share, sorry, I was going to share, like, we actually ended up switching all of our Sanger over to ONT. So we killed Sanger as of the first of the month, and now everything that we do is like any long amplicons or plasmids or anything is on ONT. And our user base has taken quite a liking to it, I think. Maybe a few sad souls that still want Sanger, but like, you don't have to do primer walking, or you just have this, everything's all in one read. So I don't know if anybody else had any experience or thoughts or questions.
I mean, I had a question. I don't know how, because I have not used it directly. Like, how does the accuracy compare, and is it good enough? And
Yeah, I would say on the new LSK 114, the things look great, especially of that, because a lot of things, range from, honestly, we've put up things on there from like 300 base pairs, like amplicons that small, up to like, 20, 25 KB, and the accuracy is great. We've had very good success with it on the LSK 114. That is.
Okay, thank you.
Are you primarily using just the rapid technology, or?
Yep.
Okay. Yeah, and are you competitive with the vendors that are out there doing this?
Yes, we have dropped our, because we were losing a lot of business to like Plasmidsaurus, and we were like, well, this is bogus. Like, we don't want money to go off campus, so we created it. And we basically are very competitive. We're like slightly more expensive, but we also give a very detailed report, and everything analysis with it. So it's kind of a whole thing for one. But
Yeah, we definitely looked into, because somebody started a Plasmasaurus Dropbox here as well, and so we were a little reactionary. We were trying to get ahead of it. We haven't yet, but I would be interested in chatting with you about it, Molly.
Yeah, for sure.
Because it is definitely something that we would like to offer, and we are like ONT, woo, right over here in Vermont.
For anybody that knows Scott, yes.
Yeah.
We're actually we're going to offer it too as well on our Pack Bio, but for polyploids. We think like the long amplicon will do better for like on Pack Bio, just with a little bit more accuracy as well. So that we're going to switch that over as soon as we can. So, let's see.
So I had another question if nobody has. So for long read, like generally what is the proportion of people who want to do DNA, like whole genome or transcriptome, and transcriptome for isoforms, etc.? And are they still mainly like looking at 10x paired with either ONT or PacBio? Because or are they looking at other? Is there interest for other things to be compatible?
Well, I can say at Wisconsin here, I would say most of our long read business is DNA focused, less transcript or RNA focused. We do have some, it's just, I would say like our proportion is like 80-20 DNA to RNA.
Okay, but is that because of what is offered and available, or is that because of what people want?
I think it's kind of a mix. I think because we offer everything that we offer everything that we have available to us, but I do think that the DNA stuff, I think, just kind of is a little bit more fleshed out.
Okay.
If that makes sense.
Yeah.
And DNA is better than RNA. So you didn't hear me, I didn't say that. But I think it's just RNA can be finicky. And this is all strictly my opinion, but so
okay.
I don't know if anybody else has experience one more than the other. Sequin, do you guys?
We do more RNA on our side just because the nature of request, right? It's whatever people need, right? We have to serve whatever product that come in. So we do more RNA, sometimes a lot of just isoform discovery, right? We want to see the full length. And sometimes I think we have one product doing RNA methylation. Check modification the RNA directly. So
Okay. And do you all mainly use like 10x, like single cell, or MassSeq or IsoSeq going into
Well,
this, or do you
yeah, in
use
our
other?
in our case, we have a couple of the 10x data with the
Okay.
OT data, just to match cell barcode, right?
Yeah. Yeah.
Yeah, so you can do long reads plus the short reads.
Sure. Yeah. Mhm.
Does anybody have any tough organisms that they're not able to get DNA or RNA out of? We can extract everything from everything. This is awesome. I might have to hit some of you guys up. I'm just kidding. I think we have I think fungus, I think, is still that weird thing for us. We've had some success, but then you get the DNA and then it doesn't want to work. And you're like, well, but all my QC metrics say you're fine, but you're not. So I think that's where I would like, I don't know, somebody has to have like a magic solution out there, right? Where it's like a fourth QC check. Because like we check spec, we check to see if it's clean, we check the quantification, we check the size, but there just seems to be another QC metric that we just can't identify that needs, because like all those check out and we're like, but this should work. And it's usually fungal, or we had this cold water, like Antarctic fish that we could not sequence, or like the DNA looks great, but we couldn't sequence. So if anybody's got any like tricks, we've tried. I mean, I hate to admit it, we had to like kind of give up because it just wasn't working.
Is that, do you struggle on every platform with that?
Yep, we tried. Well, I mean, every platform meaning direct. So like ONT and PacBio, it does not work. Illumina, it works fine because you PCR the crap out of it.
Yeah.
It works. So
we ran into an issue. We had some fish that we've been sequencing, and we run into an issue where on ONT, the flow cells just crash. So we just kind of forced it through, and we're like, all right, we're not getting enough sequencing. We're going to throw it on another flow cell and another flow cell and another flow cell. We did try, it did help a little this seems ridiculous, to sonicate the samples a bit, because we had really nice big DNA, right? Huge, long. And we're like, okay, is it just that, they're getting in there and it's just getting stuck in the pores and not able, maybe there's some pseudo knotting happening and it's not being able, it's just getting stuck in the pores, and we just could not get rid of it. We couldn't get them to sequence all the way, like you said. And so we're like, maybe if we have shorter length, and you don't want to do that, but at the same time, sometimes you got to do what you got to do.
Yeah,
and so we've
like
been
the worst.
yeah. It seems so silly to chop up your DNA per long read, but
yeah,
it worked a little. Not, it wasn't amazing, but it did seem to help. And I mean, it was like, we just have this little micro sonicator water bath thing, and it's like zap it for 10 seconds or 20 seconds.
Interesting. Yeah, like we tried like needle shearing digestions with whatever, and it just, I don't know. Yeah, we even sent it to a company. You guys heard of, I don't know, Purigen? They do like they have got like a little machine that does like DNA extraction, and we sent it to them and we were like, help us clean this DNA, and even that didn't work. And I mean, I was very hopeful, but nothing against them. They made other things look good. It was just this one sample, but yeah. Does anybody here have a Revio yet? Me either.
Got that news about, we don't have one here yet. I'm putting a testing proposal for that. And hopefully, I mean, for the first round, you already don't get it. But I have my hopes still. But I think right last week or earlier this week, there is a new announcement from PacBio. There is a new financing kind of option available.
Oh,
Yeah, something I think PacBio is pairing with a finance company to offer. There is no any detail in that announcement, just 6 months as a cash, something like that. It's like typical financing language. So I want to find out some details about that option. Don't know whether, Sarah, you have anything.
Yeah, it's called PacBio Capital. I think we have a company that will help you sort of finance a system. Here, I'll send you the website. I don't really know much more than what was on the press release, but I can find out more if people want to get more details. You can just email me and I'll connect you with the right people.
Okay.
I put my email in the chat if people want to reach out.
Great, thank you.
does anybody know of any good conferences like coming up that? I mean, I know ASHG is a big one coming up. We don't typically go to that one, just because it's big and we don't do a lot of human work. But anybody else have anything big coming up that kind of showcases any long read stuff? Well, I'm going to put in a shameless plug if anybody wants to come to the ABRF Midwest meeting. It's in Ann Arbor, and it's going to be really fun. So, because I'll be there, Sequin will be there.
Also
be
put in
there.
a plug for Nerds,
There you go.
which is next month in Burlington. Height of
Awesome.
leaf season.
That's a good point. They need to give us more than 30 seconds warning, by the way,
Yeah.
that this is closing. But for real, it was good again this time.
Yeah, let's talk at Midwest when you're
Okay.
here. Come.
Kevin now, so we can't complain.
Well, yeah,
Yeah.
I guess 15 minutes wasn't enough additional time either, was it?
No,
That's okay.
I think this topic is passionate to so many of us that I think we could talk for weeks and still there wouldn't be enough time. So thank you all. And so let's kind of just go through the different topics and talk a little bit about. I'm just going to highlight. I was in the short reads session and highlight some of the key topics that were discussed in your sessions, and we'll go down the line. But really, in the short reads session, it was all about the money. Folks were why are we looking at going? We have an Illumina system, we have a NovaSeq or some. We want to sequence our smaller projects at the same cost that it costs to feed the higher throughput Illumina systems. And so that's really one of the major reasons why folks are looking at that, looking there at the. There was some discussion on working with low diversity libraries. Didn't seem to be an issue with the Element. Some are going with, instead of doing the 5% recommended, 10% is what they're going with instead. Then also the same way when you're spiking in Phi-X for bisulfite conversion. So hopefully I've covered the major highlights, and we can move on to the next topic. That would be the